Hey everybody, my name is Adam. I am an MCAT tutor and top MCAT scorer, and I'm going to be walking you guys through an MCAT biology passage today with Shemmassian Academic Consulting. So let's dive into it. So first, where in the MCAT would you find a passage like this, right? So there's 4 sections to the MCAT. Remember, MCAT stands for Medical College Admissions Test. You're going to take this exam prior to applying to a medical school program or another type of graduate school that would require the MCAT. And it's 4 sections. Each section is 95 minutes long, except for the second section, which is 90 minutes, right? Which gives you a total of 6 hours and 15 minutes of testing. So first section is going to be your chemistry and physics section, where they test you on that. Then you get a short break. Second section is going to be CARS, which is critical analysis and reasoning section, which is just reading passages and answering questions based on what you read. Then you get a 30-minute lunch break. Then you go into the third section, which is where we are today, which is the biology section, where you're going to do biology and biochemistry. And then you go into the final section after a short break called the psychology and sociology section, kind of testing that side of your knowledge as well. So in this third section that we are in today, the biology and biochemistry section, we're going to have 95 minutes, and we're going to have 10 passages and 15 discrete questions. So a nice strategy to start out with if you're just taking your first full length is to do those first 15 minutes and do the 15 discrete questions, right? Skip to those, the ones that are not associated with any passage. So you take about 1 minute per question and you do those first 15 questions. So now you have 80 minutes left for 10 passages. So you go back to passage 1 and you take 8 minutes and you do that passage. Passage 2, also 8 minutes. You want to do 8 passages all the way through for the 10 passages, and then you get about equal time to give yourself a good chance to answer each question to the best of your ability by doing appropriate timing. So that's a good place to start, and then as you start to practice full lengths and practice passages on your own, you can adjust that strategy to what works best for your strengths and how to account for your weaknesses in a way that can make you a strong test taker. So let's dive into this. We'd want to do it in 8 minutes normally. I'm going to do it a little bit longer than that today because I'm going to be breaking everything down in my total thought process as to what I'm thinking as I'm answering these questions and as I'm reading this passage and what I'm highlighting. But yeah, when you practice this on your own, like if you want to do this before I go through it, do it in about 8 minutes or less, and then you can watch my breakdown. And we can go through it together. So let's dive into it. So we're going to start with the passage here. I'm going to pull out my highlighting tool. If you're on like a site, an MCAT site or something, you can use, if you're on a Mac, Option H would let you highlight like Option and H at the same time. If you're on a Windows computer, it'd be Alt plus H, so holding it, so Alt then H, and then you can highlight your text and it will actually highlight it in yellow. But I'm on a whiteboard here, so I'm going to actually use my cursor. Okay, so let's dive into this. Let's highlight some of these terms as we read along. So propiomelanocortin, POMC. So I'm going to highlight that because it's an acronym. It's good to be able to find those in case we need it. Neurons in the arcuate nucleus of the hypothalamus, ARH, right? So I'm going to just highlight ARH because I want to highlight this whole sentence, but that's arcuate nucleus of the hypothalamus. Control energy homeostasis by sensing hormonal and nutrient cues and activating secondary melanocortin sensoring neurons. Okay, it's just a lot of words telling us a little bit what's going on. I'm going to highlight homeostasis, right? Just like maintaining that middle set point for the body. Researchers identified the expression of a G protein coupled receptor, GPR17. Okay, I'm going to definitely highlight that because we want to know, we do know, right? You can see that as kind of a clue. G protein coupled receptors are a piece of outside knowledge that we know from the AAMC is something that we should know about, right? So we're like, okay, they could easily test me about this, so I'm going to highlight that. In the ARH, we know what that is, and hypothesize that it contributes to the regulatory function of POMC neurons on metabolism. Okay, so this guy has to do with regulation, this G protein coupled receptor, regulation of metabolism in these particular neurons. Cool. Let's go on. In order to test this hypothesis, researchers generated POMC neuron-specific GPR17 knockout mice. Okay, so there's another acronym, which is the neuron, the G protein coupled receptor, and then the knockout, right? So they're knocking out this receptor, which is supposedly regulatory. I'm also going to highlight the word researchers because, right, typically in an MCAT biology passage, there's going to be kind of the setup, which is this first paragraph, and then they'll begin some kind of study where they test the theory that they have, right? And that's why I usually highlight the word researchers, or you could highlight the word test, right, or something like that, to kind of let you know, in case you do have to refer back to the passage, where they started their experiment. So in this case, it's the second paragraph. It's very early on, but sometimes there will be multiple paragraphs of explaining like the theory behind things first before they start their study, or there will be multiple studies. So anyways, so researchers, they made these knockout mice, okay, and they determined their energy and glucose metabolic phenotypes on normal chow diet and high-fat diet. Okay, so there's two different diets, so I'm just going to highlight them. Right, so two different diets. In experiment 1, these same researchers measured body weight and weight gain of the wild type, which that's the normal chow diet, and the PGKO mice on a high-fat diet. Okay, so they have the two different types of mice on high-fat. Body weight was assessed at 5 months and 10 months after the diet change for adult mice. The results from this experiment are shown in figure 1. Okay, so this is experiment 1. And let's look at the graph briefly here, right? So we got body weight and weight gain on our y-axis. We have time on the diet over here, and then here we have the types. And again, we have the wild type and the knockout one again. So we want to look for these asterisks, right? These asterisks mean that they're different, or basically that the thing worked, right? So we see an asterisk here over the white box, which we know is wild type. So wild type gained more weight, significantly more weight than the knockout. And then over here, weight gain, well, they put it in the reverse here, but it doesn't matter, right? Because these asterisks just mean that they're different from each other. It doesn't necessarily mean that one is bigger and one is lower. It means that they're just different from each other. So here we got, right, on the left, body weight, right, greater in those with the wild type, less in those with the knockout. Weight gain, same thing. Greater in those in the wild type, less in those in the knockout, right? So we have both of those things being assessed. But also important to note, 5 months of being on it, being on that special high-fat diet, no difference yet, right? They're the same. There's no asterisk here on this 5-month mark. Only after 10 months do we see a change, right? And then this graph over here is just showing the weight gain. This would just be overall, right? This is not the 5 months, not the 10 months, just be overall, right? Because we don't have a time thing for both of those. All right, let's see what else we got. Neuropeptides from POMC neurons are known to play a role in appetite regulation. Alpha MSH is anorexigenic, right? Okay. While the effect of beta endorphin, beta EP, on satiety is context dependent. Okay. The bioavailability of alpha MSH and beta EP is partially determined by the expression of POMC neurons and subsequent proteolytic processing, PC1, PC2, CPE. Right, they're just giving us a lot of words here. I'm going to highlight them so I know they're there, but we don't really know what this means yet. Researchers measured alpha MSH, beta EP, and POMC neuropeptides in the mediobasal hypothalamic samples from mice fed high-fat diet for 2 weeks, right? So they just measured all of this stuff on the high-fat diet for 2 weeks, and then here's our results, right? So we got alpha MSH per milligram, right? So in the knockouts, we see that they're different from each other, male versus female. Okay, so now we're looking at a completely different thing here. Interesting to note, right, there's no difference in the males between the wild type and the knockout for alpha MSH. But there is a difference in the females between wild type and knockout. And then there's difference on both between male and female, but it's the same, right? You see the 3 asterisks. So it seems like, right, so you see wild type versus wild type, significant male versus female. Knockout versus knockout, significant male versus female. The only like significance within we have is for female mice. Okay, so that's interesting. That's probably hormonal related. And then we go over here, we got beta EP, which is again the opposite, context dependent, right? No difference on the males within each other for the both. And then there is, again, a difference within the females, and then a difference between male and females only for the knockout. So, kind of interesting there. We'll see what kind of questions they ask us about this. We don't need to fully understand everything. We just need to kind of know what they're looking at and see if they ask us any questions and then dive in a little deeper, because we don't have enough time to fully evaluate everything when we read through a passage. Okay, so before I dive into these questions, I want to talk a little bit from our content guides at Shemmassianconsulting.com/MCAT, and kind of dive through a couple figures that are related, so we can kind of get a basis for some of the MCAT content knowledge, some of the high yield MCAT content knowledge that is tested on similar passage like this. So this is from one of our content guides, and here we have a guide, like a little image here on G protein coupled signaling. So this is kind of showing at the beginning here, we got our resting state of a G protein, an internal G protein. We have our outside and inside of our membrane. Here's our G protein. Then we have our alpha, our beta, and our gamma subunits. So they're just kind of all sitting there. Nothing is really going on. And then we have a ligand coming in to bind, right? And it's often a peptide hormone, right? Because remember, we have our peptide hormones, we have our steroid hormones, and we have our tyrosine derivatives. Peptide hormones are very polar, which means they can't go across the membrane, right? So because they can't go across the membrane, they have to bind to something on the surface, right? So we're going to bind to this surface receptor, which had an internal processing thing called the G protein coupled receptor, right? So as it comes in and binds right here, we can see how we have some conformational change going on here, where GTP, right, these little like kind of bird-shaped things, are coming in and replacing GDP, right? You can see these arrows. GTP comes in, GDP is coming out. So we're going from inactive form, which is GDP bound, to active form, to GTP is bound. So when that GTP binds, we then have our alpha subunit dissociate, right? Which is what we see here, right? You see the alpha subunit over here, we have our beta and our gamma over here, right? And now that our alpha subunit has dissociated and we have GTP bound, or our active state, we can start our signaling passage, right? There's going to be a whole bunch of, we call it cascade, right, of secondary messengers to eventually do the thing that we want to do. If we want to impact transcription, if we want to impact translation, whatever this peptide hormone was signaling for. So there's a lot of different types of internal signaling, but this G protein receptor, we can have, activating, we can be inhibitory, we can be just neutral, causing a cascade of neutral events, right? So, but our alpha subunit is going to be our thing that's dissociating. Then as it does its thing, we'll have some kind of thing come back and activate our GTPase, right? Which is going to cause GTP hydrolysis, which we lose that inorganic phosphate from GTP, which then would turn it into what? GDP, right? Because GTP means guanine triphosphate. GTP means guanine diphosphate, which is just the amount of phosphates we have bound on the end of our guanine. So then hydrolysis, right? We're losing that inorganic phosphate. And now what do we have bound? We have GTP bound, which means our G protein subunits, our alpha, our beta, and our gamma, are going to reassociate, right? And when they come back together, that's going to cause our ligand to dissociate as well. So, right, they're going to come together. Our ligand here, right, our peptide hormone, is going to leave. And then we're back to the GTP bound resting state, right? GTP is bound in the alpha subunit, and we're not doing anything. So that's like our really common mechanism for our peptide hormones, right? Because they are so polar, right? So again, our steroid hormones are nonpolar, right? So they can go right across that membrane and they can make changes directly within the cell. They often don't need this type of signaling cascade. So we have a short list here from the content guides of a few of our peptide hormones. These are some of the highest yield ones. Acronym over here for our anterior pituitary is flat PEG, right? If you ever have questions about where they are. So FLAT, right? The first letter here, and then PEG, prolactin, endorphins, and then growth hormones that's actually one. It's on two lines, but it's one, right? So flat PEG, that's what it is there. And then, right, posterior, we got ADH and oxytocin, PTH from parathyroid, right, gland, it's pretty straightforward there. And then we have our two kind of highest yield, kind of opposing, it's probably our highest yield hormones on the entire test, glucagon and insulin, right? They're both peptide hormones, right? So a nice way to remember which one does which is, right, insulin, it ends in in, and then glucagon, it ends in gon. That tells us a little bit about which one does which way, right? So we're both talking about our storage of our carbohydrates, right? So insulin is going to want to store our carbohydrates, insulin, as glycogen, right? So we're going to be increasing glycogen, increasing our storage of our carbohydrates in the form of glycogen, which is a chain. Which means we're going to be decreasing like our serum or our blood carbohydrate levels. So that's like rest and digest, right? We just ate a whole bunch of carbohydrates, we need to store them. We release insulin, right? And then glucagon is going to do the opposite, glucagon, right? We want to take those glycogen stores, we want to stop storing them, we want to make them gone, we want to get rid of them. So we're going to take those glycogen storages, break them down, right? What's that called? Glycogenolysis, right? Lysis, breaking down. So glycogenolysis, glucagon is going to stimulate that, which is going to increase our carbohydrates in our blood, our sugars in our blood. Yeah, so those are opposing hormones, both released from the pancreas, both peptide hormones, right? So they're going to have some kind of symptom, kind of like a GTP or a signaling cascade, a G protein signaling cascade, something similar to that. And then some of the other opposing ones are calcitonin and PTH, another high yield opposing thing. They have to do with, instead of sugar in the blood, they have to do with calcium in the blood, right? So calcitonin, right, just like with insulin, calcitonin, it has an in at the end of its name, which means we're going to want to store our calcium. Where do we store calcium? In our bones, primarily, right? So we want to release calcitonin. We have too much calcium in our blood, and we want to store it as bone, right? Stimulating those cells within the bone that make blood, or that make bone, called osteoblasts, right? Osteoblasts make bone, BB, right? So blasts build bone, BBB, pretty straightforward, calcitonin. And then if we don't have enough calcium in our blood and we need calcium, right, to do skeletal muscle contraction, we're going to release the other hormone, which is PTH, right? PTH is going to stimulate those osteoclasts, right, which are the opposite of osteoblasts in our bone. Osteoclasts, we have that C, it sounds very like coarse, right? They're breaking down the bone, they're cutting. Osteoclasts cut the bone, break the calcium out, cut calcium out of the bone, clasts, cut calcium, put it into the blood. So right, PTH is going to do the opposite of calcitonin and get us our blood levels of calcium back up, but it will do that at expense of our bones, right, making them a little bit more weak. And then a final thing I wanted to just quickly note while we're talking about hormones is some of the higher yield things, again, with hormones, is our three axes, right? HPA, HPG, and RAAS. They're all stimulated from different things. I guess some of the most important things to remember is, like, when do we use these, right? So HPA, what you want to remember with HPA is stress, right? So what's happening, our trigger, right, or the thing that causes the axis to turn on is going to be stress. And then what's our end thing that's going to be released? It's going to be from our adrenal gland or our adrenal cortex. It's going to be cortisol, right? So we're going to have stress is going to be our trigger for this one, releases cortisol, which is going to help our body cope with that stress in different ways, right? So it does a couple different things there, cortisol does. And then HPG axis, right? Notice how it's pretty similar, hypothalamic pituitary, but now we have a gonadotropin, which means we're now going to our gonads, right? You see that word gonado at the beginning, right? You think, what are our gonads in males, right? Testosterone are like in our females, right? We have our two different hormones, estrogen, progesterone, right? So we're going to go to our gonads, and you think, what is our trigger for this, right? We're going to have kind of entering adolescence, right? Entering puberty. And then what are these hormones that are released there going to cause? They're going to cause our sexual changes, right? Our secondary sex characteristics, right? Developing, right? All the things that happen during puberty, right? So puberty is going to happen, and then we have our sexual characteristics. And then RAAS, right? Renin, angiotensin, aldosterone system, right? Our third kind of high yield hormonal axis, right? Renin's released from the kidneys in response to low blood pressure, right? We want to increase that blood pressure because it's too low. Renin's released. It's actually the enzyme that converts angiotensin into angiotensin 1 and angiotensin, and then eventually to angiotensin 2. Angiotensin 2 will help us release aldosterone, which is going to be our main hormonal regulator of our our filtration kind of within our kidneys. And it's going to happen in two different places, right? The last two places, right? The distal convoluted tubule, are called the DCT, and the collecting duct, right? It's called the CD, right? So that's going to be the RAAS. And when those are released, right, it's going to cause us to, right, aldosterone has to do with salt, right? So it's going to help us retain that salt or get more salt into our blood serum, which, again, water follows salts, right? We talk about osmosis. You look at that in some of the other content guides talking about osmosis. Water is going to follow solutes always. So if we retain our salts, our different solutes, water is going to follow that, which is going to put more water, more liquid into our blood, increasing our blood pressure, getting us back to where we want to be, right? So those are kind of our three triggers, right? We want to think blood pressure too low, BP too low, we cause the RAS, which cause our blood pressure to go back up because we released aldosterone, helping us get back to a regular blood pressure. Okay, cool. So that's a little bit of the content there. So now let's dive into these 5 questions, and let's see how we do. Okay, so hopefully you remember a little bit about what we went through on that passage, but we'll kind of go back to each part as we get to it. So number 1 here, so researchers hypothesized that PGKO, right, that's our knockout mice, maintain a low weight on the high-fat diet by increasing beta oxidation, right? So we should know what that is. If this hypothesis is correct, which molecule might they expect to find at higher levels? So what will we have at higher levels than the wild type if this theory is correct? So if we increased beta oxidation, that means we're taking our fatty acids and we are producing acetyl-CoA to get transferred into our citric acid cycle to generate energy to maintain that low weight. Okay, so let's look for that. Okay, so pretty straightforward right away. We're looking at it. We see the product of beta oxidation right away, acetyl-CoA. But let's talk through the other answers and make sure that they're not correct just for the sake of practice. Pyruvate, right, that's going to be our terminal molecule of which pathway it's going to be glycolysis, right? So glycolysis is so that's going to be carbohydrates. We're talking about the wrong thing. We're talking about fat diets here. Pyruvate is the terminal thing when we have carbs or sugars. Glucose 6-phosphate, right, one of our things in glycolysis. Glucose 1-phosphate, again, one of the middle molecules in glycolysis, right? So these are all carbohydrate or sugar-based, right? So nothing to do with fat or beta oxidation. Acetyl-CoA has to do with both fat and beta oxidation. So B is our only good answer there. All right, number 2. Based on figure 2, which of the following conclusions is true? Right, remember when we were looking at this, the only thing that we could really notice is that there's a difference between females and males, right? That's the only thing that happened on both of them, especially with the knockout, right? The knockout had a difference between females and males in both alpha MSH and beta EP. But let's see what the question's going to be here. Okay, which of the following conclusions is true? Okay, so they don't really give us much. We have to go through each of them. So females have higher levels of POMC neurons. Right, we didn't really look at that, right? We looked at not POMC neurons, but alpha MSH and beta EP. So didn't look at that. So we're gonna rule that one out. B, the knockout mice produce higher levels of neuropeptides. Let's see. Not always, right? Because between wild type and the knockout, we only have differences in females, but we don't in males. So that would not always be true because they would have to quantify that that's only true in females. It's not true in males. So B is not true. All right, let's look at the next one. C, wild type females have higher levels of alpha MSH than wild type males. We'll see if there's an asterisk between that. It's a very specific statement, so we can prove that pretty quickly whether that's true or false. So we're looking for alpha MSH, right? That was the first graph, and we're looking for wild type females versus wild type males. So let's go over alpha MSH, right? This is B, graph B. Wild type females, wild type males. We have 3 asterisks between them. It is significantly higher. Okay, so we know that C is true. There is no way to refute that. So definitely C is the correct answer. Let's just disprove D here, make sure it's not true. Wild type males have higher levels of intracellular beta EP than wild-type females, right? So we're going to look at the second graph, we're going to look at beta EP, and we're going to compare wild-type males versus wild-type females. All right, let's look at this here. Wild-type males have lower, right? And also there seems to be no difference, right? There's no asterisk between the wild-type on the beta EP. So no difference, no asterisk at all. And even if it was, the females are a little bit higher, but not significantly higher. So even if the statement was flipped, it would still be false. All right, so cool. Now one's C. Let's look at 3 now. Researchers are interested in studying a new G-protein coupled receptor called G-protein coupled receptor 84. Which of the following mutations may researchers introduce to the protein to decrease GPCR84 signaling? Okay, so we're looking for decreasing GPCR84. So when it comes to MCAT biology questions, it's very common that you're going to get something around G-protein coupled receptors, and also something where you basically just have to recall your outside knowledge, right? This is not really based on the passage at all. So we would call this like a pseudo-discrete. It's based on the passage, but there's really, the only thing in the passage is just mentioning that a G-protein coupled receptor exists. There's nothing about that would tell us how this works. So yeah, when it comes to MCAT biology questions, we need to be prepared for a content question like this. Okay, so how do we decrease GPCR84, right? And if we go back to our content guide here, remember resting or GDP bound is when we don't have anything going on, and then active is when we have GTP bound, right? So we want to figure out how to increase the GDP bound or decrease the GTP bound in order to answer this question, because right, we want to decrease the signaling. So let's see, we got A, mutate the alpha subunit so it is locked in GDP bound form. That's what we want. So A is definitely correct. We don't need to evaluate the other ones. On test day, you shouldn't. You know this is correct. Move on. But let's, for the sake of practice, rule out these other ones. Mutate the alpha subunit of the G-protein coupled receptor so it is locked in GTP bound, right? That's going to be always active, right? So that would increase, right? So that's the wrong direction. So we'll X that one out. Add an agonist ligand that binds to G-protein coupled receptor, right? So an agonist would make it activate, right? An antagonist would make it inactive, right? Those are our different types of ligands. So agonist would cause more activity, not decrease. If it said antagonist ligand, that would also be correct, but right, we're never going to see 2 correct answers. It's not how the MCAT works. So agonist is wrong. And then D, expressed G-protein coupled receptor on cellular surfaces that are spatially close to agonist ligands, right? So how do ligands work? They have to actually go there and bind. So if we get close to it, it's going to work, right? So if we're spatially close, it's going to work, which is going to increase our G-protein coupled receptor signaling. So B, C, and D are all going to cause increases. So none of those are good answers. We already knew that A was correct, but luckily through going B, C, and D, they also were definitely wrong. Okay, let's look at 4 here. Researchers observe a surge in alpha MSH immediately after eating, along with which other hormones? So what other hormones are released after eating, right? We want to think rest and digest. We want to be thinking a storage hormone, right? How do we store something, right? So insulin, right? We want to store that. So that looks good. We're going to stick with A for now. We'll see if there's a better answer. B, glucagon, right? Gon, that's going to be a breakdown hormone. It's going to get rid of stuff. So nope, not good. Somatostatin, right? Doesn't really do a whole lot in our body except for inhibit both insulin and glucagon. It's our other hormone released by the pancreas. But no, not a good answer, right? Because it's only going to be inhibiting the 2 of these, and because we just ate, we're going to increase one of them, which it's insulin. That's the one we're going to increase. So somatostatin, bad answer. And then norepinephrine, right? That's going to be a stress hormone. Right after eating, our body is the opposite of stressed, right? We now have just eaten. Now our body's like, great, now I have energy that I can store and I can use later. So norepinephrine will probably decrease, but even so, like, it's definitely not going to help us in storing stuff that we just ate, right? It's going to help us in like surviving a bear attack or surviving something where we need to kind of activate those different types of things. Okay, so good, we got that one. Now number 5 here, zoom out a little bit so we can see the whole question. In the high-fat diet, researchers feed mice unsaturated fats, okay? which of the following represents a fat with the most double bonds? Okay, so we just got to find the one that is going to have the most double bonds, right? Remember, so unsaturated just means we have a double bond. Saturated would mean we have no double bonds. Unsaturated means we have at least one. We could have, the entire thing could be double bonds, right? So we don't know how many, but let's kind of look at these, as to these names of these unsaturated fats and see which one's going to have the most. So 18, 1, delta 9. So let's just kind of break down what this means. 18, that's going to be our number of carbons, right? So remember, I'm going to grab my pen here and I'll draw out a fat here, right? So we're going to have our head of our fat over here, right? It might be, I don't know, carboxylic acid or something like that. And then we have a chain of carbons like this, dun, 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 dun. And if we're unsaturated, right, one of these is going to be like a double bond, like this, at least one, and maybe more. 18 is telling us how many carbons do we have, right? 1, 2, 3, where you go all the way down to 18, right? And then the delta 9 is going to tell us where our double bond is. So that's going to tell us after number 9, right? So if this one was number 9, like if we pretended like we skipped a few, this one was number 9, that's where our double bond is going to be. And the number right before the delta tells us how many double bonds we have. So 18, 1, delta 9 means we have one double bond at position 9. Okay, so then B is 18, 2, delta 9, 12, right? So it doesn't really matter how many carbons, doesn't really matter where they are, not for this question, because we're just trying to pick most double bonds. So 2, the thing that matters, so we got 1, we got 2, we know 2 is more than 1, so B is better. And then C, 28, right? Remember, we don't care. 1, right? That's low. And then position, 17, carbon 17, right? Again, we're looking at 2 is the highest so far. And then D, we got 28. 3, position 9, 17, 23, right? So we got 3 double bonds here and 28 carbons. D is going to be our best answer there for that. Okay, so cool. That concludes the passage there. That one was fairly straightforward, but a lot of content knowledge brought in there as well. So I hope that breakdown was helpful for you guys. Yeah, so if you liked what you saw, there's plenty more videos. Check out our other videos as well with other passage breakdowns. And if you could click the link in the description below, we have an MCAT question of the day that can be set to your inbox, right? So one question every single day in your email. Very helpful to help you keeping up with doing daily practice. If you just needed to get yourself going or whatever your study strategy is, an email to kind of start your studying kind of can be helpful. Get started with a question. So yeah, thanks for listening, and I'll see you guys next time.